Namaste. Welcome to TSAT channel. Today we are here to discuss degree college lecturers, PGT, TGT. I think just this degree college lecturers recruitment preparation for the subject microbiology. So, for this subject the syllabus is being given and it is uploaded. So, the number of chapters that are given are 9. Myself, I am Dr. K. Anuradha from Bhavans Vivekananda College. I am heading the department of microbiology. Along with me, my colleague Dr. Aparna is here.
Namaste.
We both will be handling this paper today. For your preparation, I am sure this session is going to be very useful for the subject microbiology. Coming to the syllabus for microbiology, we have the first unit, general microbiology. For any topic to start with, the history of microbiology will be the first fundamental topic, wherein we will be looking into the contributions of various scientists, especially for microbiology, Pasteur's contributions, Robert Koch's contributions, and Louis Pasteur, and we have Edward Jenner, and all the eminent scientists' contributions. You have to be knowing it because they may ask you questions from the contributions of scientists. Then coming to the other topics, the second important topic is microscopy. So, here you will be studying about light microscopy, bright field microscopy, that is light microscopy, and dark field microscopy, fluorescent microscopy, and electron microscopy. Here most importantly, the principle of the microscopic techniques and also for what purpose you use, that is the applications. Say for example, for all live organisms you go for dark field microscopy, and for observing the various specimens like algae, fungi, bacteria, light microscopy. So, fluorescent microscopy for understanding the internal structures, and electron microscopy, both SEM and TEM, for topographical observations, and for scanning for internal specifications. You go for all these different microscopes. So, the importance of microscopes, you don't leave it, you please emphasize on that. Then coming to the microbial cell, because it is exclusively the microbiology paper, so all the discussion refers to prokaryotic cell. So, taking reference as bacterium, so study in detail the internal components of bacteria, especially emphasize more on bacterial cell wall, that is about the peptidoglycan content, differences between gram positive and gram negative cell wall, and what is the main differences between these cell walls, and also the special cell wall structures of archaebacteria and also actinomycetes and Mycobacterium tuberculosis. You learn about the important features of cell wall, and also emphasize on all internal components like ribosomes, what are the types of ribosomes, mitochondria, and of course you talk about in the eukaryotic forms, and here it is only for prokaryotes. So, look into all the details of the cellular components of the prokaryotic cell. And coming to the sterilization techniques, both physical sterilization and also the chemical sterilization, you please study clearly, because in physical sterilization you have heat as a component, dry heat, moist heat sterilization, and also autoclaving, and principles of all these sterilization techniques, and the other physical sterilizers like bacterial filtration also. Filtration is also very important because the cultures are sterilized through filtration process, and the role of the membrane filters, nitrocellulose membrane filters, and all you have to consider, and most importantly the principle of sterilization, and also the what are the various articles and objects and media, what sterilization method you use. Say for example, for autoclaving, media preparation, you sterilize media using autoclaving. So, that way you have to be looking into it, what are the what are the articles, and what are the glassware, media, which method of sterilization you will be using, you have to look into it. Then the next important topic of this is media preparation, that is growth. Of course, before you get into the growth characteristics, media preparation is also very important, because different types of media you will be using for different types of bacterial cultures. Say for example, for Vibrio you use VR medium, Venkatraman Krishnan's medium, and enrichment medium, tetrathenoate broth, alkaline F broth. And for enriched media you go for blood agar medium, and you use egg based medium, and also learn about differential media and also selective media. When you are preparing for selective media, you should also know what is the inhibitory agent you are adding in it. Of course, some of the differential media are also what you call come under the selective media. So, for example, McConkey media, when you talk about differential, it is both for Enterobacteriaceae members, lactose fermenting, non-lactose fermenting, you will be able to differentiate. But at the same time, you will also quote for the selective media also. At that time, you need to know what is the inhibitory agent you are adding in it. So, such minute aspects also you have to concentrate, that is the sodium chloride what you use as for omitting all gram-positive forms. So, what I mean to say here is, when you talk about enriched media, what is the component you are enriching with? When you are talking about selective media, what is the inhibitory agent you are using it? For Mycobacterium, what selective media you are using, like Lowenstein-Jensen media. So, all about in media components, the carbon source, nitrogen source, energy source, as well as all the examples for these media you have to be learning, so that it becomes very easy. Make a nice tabular form out of it and try to cover all the bacterial cultures. Then the growth curve and the measurement of growth, with reference to this, you need to know what are the different phases of the growth, and also the calculation of generation time. Say for example, for E. coli, the doubling time is just 21 minutes. But considering that, you also should know how you calculate the generation time. Sometimes they may be giving certain data and ask you to calculate the generation time. However, the growth constant and all the other parameters which you need to consider, but simple mathematical solving can also may be given, taking log values into consideration, that it equalizes to 1 or 10 like that. Okay. So, in growth characteristics, features of growth characteristics you learn, and here how you culture the organisms, like especially the continuous culturing. You need to emphasize on chemostat, turbidostat, all these aspects, so that you are able to answer it, whatever that comes from the chapter growth and its characteristics. Not only that, even the physical factors they may ask you. So, try focus even on the physical factors of growth. Then as I said, synchronous culturing and continuous culturing methods you have to also focus. Then bacterial sporulation is another concept. Here the genetics of bacterial sporulation, what is the chemical composition of a bacterial spore, dipicolinic acid content, all that you need, and what are the examples of gram positive forms you give, all that you try to cover it in bacterial sporulation concept. And the nutrition is also one important aspect. Here you may be asked many questions, because the nutrition covers both autotrophy as well as heterotrophy. And not only that, again you will have chemolithotrophs, lithotrophy, chemolithotrophy, organotrophy and all. So, mixotrophs and methanotrophs, please, methylotrophs, all that you take examples of it, in what way it differs from the other nutritional types. When it comes to nutritional types, as I mentioned, carbon source, nitrogen source, you have to be very clear with it, so that your classification becomes easy, and you will also understand their metabolic activities. Then there is another simple topic which is kept over there, that is the algae and fungi general characters. Here when you are reading this, because all that what you are referring, the earlier topics are with reference to bacteria. The last topic is that is algae, fungi. Here the classification is very important. Classification of algae and fungi and economic importance. That too, whatever examples we are referring to microbiology, please keep in mind about mushrooms and all other things. So, in this first unit is very, very important. If you are sound in this first unit, it becomes very easy. All applications of microbiology, like food microbiology, medical microbiology and environmental microbiology. Because this fundamentals, the same basis you will be applying for those branches of microbiology also. Okay. So here we have come up with a certain model question. Then what does the HEPA stands for in HEPA filters for laminar airflow? So as I was mentioning sterilization methods, HEPA stands for what? So various A alteration is given over there. So the correct answer is high efficiency particulate air filters is the correct answer, because in and out every day you will be using it in microbiology lab, these laminar airflow. Then they may be asking you match the following also. You may be spending little more time when you are actually attempting such questions like match the following. Here when you see, first you see column A and column B. In column A what is written and exactly match it with column B. Continuous culturing, you have to actually match with the correct answer, that is the chemostat. Preservation technique is a lyophilization. Sterilization is sterilization. Pure culture concept refers to streak plate method. So like that, A2, B1, C4, D3, like that you have to match exactly, but spare little time and give the correct matching. Then the next question is, next unit is virology. Dr. Aparna will throw some light on this aspects of virology topic.
Yeah. Hello everyone. So hope your preparation has started and you are in good pace of preparation also. So today I will be discussing about the important concepts that are there in virology. So first let us take a look into what aspects are there in virology, or what syllabus is prescribed in virology. As we all know, virology, it is a study of viruses, and in virology the important concepts that are mentioned are characteristics and classification of bacteria, characteristics and classification of plant and animal viruses. So when we spell the word bacteria, viruses that infect bacteria are called as bacteriophages. So when you are studying about the characteristics of viruses, what I suggest you is, you don't just stick to the characteristics and classification of bacteria, animal and plant viruses, but you just understand a little bit more of that things, because when you are speaking about classification, in classification we have to look for LHT system of classification, and they may ask you a question, what is a classification which is based on molecular mechanisms? Then it is Baltimore classification. So there are it is categorized into 6 groups, Baltimore classification. So look into those things in very much detail. The next type of advanced classification system is ICTV classification system, that is International Committee on Taxonomy of Viruses. So they have grouped the viruses based on the one that are infecting animal, the one that are infecting plant, the one that are infecting bacteria. So when it comes to bacteria, you call them as bacteriophages. Similarly, there is an updation in the ICTV classification system also. So what are those updations? There are certain types of viruses that have been discovered which are not only affecting these 3 which are mentioned in the syllabus, but they also affect cyanobacteria, which we call them as cyanophages, actinophages, which infect actinomycetes, and there are certain types of viruses that even infect fungi. We call them as mycophages, phycophages. So look at those terminology also. And since the characters is mentioned, so you need to study about the characters of the viruses, like you study about its structure and classify them, whether they are enveloped virus or a non-enveloped viruses. List down some examples of what are enveloped viruses and what are non-enveloped viruses. Then you also list what is the symmetry of a virus, whether it is a cubical symmetry, icosahedral symmetry, helical symmetry, or if there is any deviations in the symmetry, that also please make a note of it and write the examples of the symmetries that are there. Okay, and you classify the viruses as DNA viruses, RNA viruses, whether they are you remember example for each and everything. Say for example, if DNA viruses are there, so what comes under double-stranded DNA viruses? What are the viruses that contain single-stranded DNA? What are the viruses that contain single-stranded RNA and double-stranded RNA? Apart from this, certain viruses, they exhibit typical genomes. That is, some of them have circular genomes, some of them have segmented genomes, like in case of influenza virus. If you take the case of influenza virus, the influenza virus has 8 segments. So that is one beautiful aspect of immunology. Okay, and that influenza virus, it multiplies with that 8 genomes, segmented genomes that are there in it. Similarly, there is one more type of virus, which is alpha-alpha mosaic virus. It is a plant virus. Okay, so like that, there are certain unique features for viruses. You please focus on that unique features. Apart from this, there are certain types of viruses which contains only protein and which doesn't contain nucleic acid, which we call them as prions. And remember the diseases. What are the diseases that are caused by prions? Like 𝗳𝗏𝗺 disease, mad cow disease, scrapie disease, so like that. And similarly, there are certain subviral particles. We should call prions and these virusoids as subviral particles. So you remember about the examples of that subviral particles also, which has only nucleic acid but doesn't have protein coat. So you remember the examples of the diseases that are caused by these virusoids, that is 𝗳𝗏𝗺 disease, that is potato spindle tuber viroid, coconut cadden disease. So such type of diseases which infect the plants. Viruses, that subviral particles that contain only nucleic acid, examples of the diseases it causes. Subviral particles that contain only nucleic acid and doesn't have a protein coat. And there are certain types of satellite viruses. Remember all these terminology when you are studying about the classification and characteristics of viruses. Now coming to the second topic that is there, the second topic is methods of cultivation and propagation of viruses. Now how to cultivate different animal viruses, because our viruses are obligate intracellular parasites, you need to cultivate them. You cannot cultivate them like how you cultivate the bacteria. So you need to understand the different types of methods that are used to cultivate a virus. So what are the different methods that are there to cultivate the virus? Say for example, animal viruses can be cultivated in chick embryo and they can be cultivated in cell lines. Okay, so there you study about some examples of the cell lines like Vero cell lines, HeLa cell lines. Remember the names of the cell lines. Okay, and next coming to the plant viruses, how plant viruses are cultivated. They are cultivated in tissue cultures. So check about that also, how plant viruses are cultivated. Then how bacterial viruses are cultivated. You check down how the bacterial viruses also will be cultivated. They can be cultivated on bacteria because they grow mostly on the host, okay, because they are obligate parasites. Now how to maintain the cultures. So you should understand how to maintain the viral cultures also, because they cannot grow in the normal medium. They require a live cell for its growth. So you need to understand how it can be maintained. Again, you need a live cells like cell lines and other things. So please go through about all those techniques that are mentioned. And then how to quantify a virus. There are some methods that are listed. So acid endpoint method, then titration method, plaque assay, POC assay. So all these different assays, neutralization assays. So all these assays are mentioned. Just list them and then you make a note of any specific virus if it is there. Okay, so this covers the first part of the syllabus. Now the next part of the syllabus comprises of type studies. So they have mentioned in your type studies about influenza virus, adenovirus, how it replicates, HIV virus, how it replicates, TMV virus. So understand the syllabus that it is framed, and each one has a unique strategy of replication. Okay, say for example, influenza virus is there listed in your syllabus. What is the unique feature of it? It has segmented genome and it is a single stranded RNA virus. How it replicates, that is the most important part of it. And if you see the case of next example that is given, adenovirus. Adenovirus is a double stranded DNA virus and it replicates by a specific panhandle model of replications. So what I am trying to tell you is, when the type studies are given, the question may be asked in your exam. Give an example of single stranded RNA virus which is present in diploid, that means in duplicate copies, then you should not struggle at that time. So, that is why the type studies, when it is given, you make a list, tabular form of it with the nucleic acid content that is there and any specific replication strategy, how it replicates when it enters, and is there any unique feature of that replication. You please make a tabular form of it, that will be easy for you in preparing for the examinations. HIV virus, it contains single stranded RNA but is present in diploid copies. So, if the question is asked single stranded RNA diploid, like how I mentioned before, you should be able to recollect that it is HIV. Okay. And similarly, there is one plant virus also that is given, tobacco mosaic virus. How the replication takes place in a plant virus? Plant cell is a different, it has a cell wall, and how virus penetrates through the cell wall, that is the mechanism you have to understand. How it multiplies in animal virus, how in animal cells like animal viruses like adenovirus which causes respiratory infection, influenza virus again it causes a respiratory infection, and HIV virus, these are animal viruses. How they have given RNA and DNA viruses, please make a note of those things. Okay. And there is one example also that is given, that is T2 bacteriophage. One bacteriophage multiplication also they have asked in the syllabus. So, it also has a double stranded DNA. A prokaryotic cell is different because you have studied that prokaryotic cell in the previous unit, general microbiology. So, there is a difference in the replication strategies. Please highlight them and make a note of it. Okay. So, very nicely the syllabus is prepared. Animal, plant, bacterial with unique strategies of replication. So, focus on those particular things and make a clear note of those things in a short nutshell form, so that you can do a capsule review when you are going for the examination. And different replication strategies of viruses, when you are studying the Baltimore classification itself, you can study the different replication strategies, how a single stranded RNA virus replicate, how a double stranded RNA virus replicate, how a single stranded DNA replicate. All those things you can study when you are studying the Baltimore classification itself. So, do not leave that classification, that you focus more, so that you can cover multiple topics with the basic things at a time. Okay. And there is one aspect called as tumor viruses. The viruses that cause tumors, that cause cancers, are called as oncogenic viruses. Both DNA oncogenic viruses and RNA oncogenic viruses will be there. How a DNA oncogenic virus will bring the transformation of a cell, and how an RNA oncogenic virus will bring the transformation of a cell. There is a unique mechanism for both the viruses when they enter inside the cell. So, please focus on those things also, and remember the names of oncogenic viruses. Okay. So, HTLV, human T cell leukemia virus, it is an RNA virus. Adenovirus, hepatitis B virus. So, all these are examples of DNA viruses. Okay. And the last aspect in this virology is interferons. So, these are called as biological antiviral agents. Suppose a body is infected with a virus, how your body responds to it. So, our body responds by secreting an antiviral agent which is called as interferons. So, read about these interferons, definitely the questions will be asked, because you are aiming for a higher exam. Do not expect the questions to be at the too basic level. Definitely when you are opting for a subject, definitely the questions will be of a little bit higher level only. Okay. Now, let us quickly go through what are the questions that can be asked, model questions. So, if you look into the first question, choose the incorrect statement from the following. Okay. So, viruses are obligate intracellular parasites, that is a correct statement. Viruses contain either DNA or RNA as genome, but never both. No virus contains both DNA and RNA. Either it contains a DNA or it contains a RNA. Next, National Institute of Virology is located in Pune. It's 100% correct. Antibiotics acts on viruses. That's a foolish thing. Antibiotics will never act on virus because it doesn't have a cell wall or a cell membrane or anything. It multiplies inside our own cell. So, when you are giving for viruses, we use antiviral therapy but not the antibiotic therapy. Antibiotics are given during viral infections only to combat the secondary infections. Don't get misleaded with such statements. Okay. So, the next question that is there is the enzymes involved in HIV replication. So, what are the enzymes that are involved in HIV replication? I told you read the replication strategies of the type studies that are mentioned in the syllabus. So, the options are Taq polymerase, DNA polymerase, RNA polymerase and reverse transcriptase. So, it multiplies by reverse transcriptase. Okay. So, these are some of the model questions that are there. You can look into any number of questions that can be there. Name the biological antiviral agent. Just now we discussed it is a interferon. So, like this you need to prepare on each and every topic some words that are mentioned in the syllabus. So, next moving on to the next topic, that is industrial microbiology.
Industrial microbiology, you know that how microorganisms are exploited for large scale production of various industrial products. You will be dealing in this topic, industrial microbiology. It's a very potential area examination point of view, because once if you say exploitation of microbes in industry, you should know what are all industrial microorganisms, whether it is bacteria or fungi. You need to list it out in for what product they are used for industrial production. And another thing is, it is very important in industrial microbiology that you screen microorganisms from the mixed microbial population, and you will isolate it and you will use it in industry. So, screening and strain development strategies are very important. In primary screening strategies, you know that how you use oxonography, how you use crowded plate technique. So, for each of these products, microbial products, we need to know what strategy to use to screen. Enrichment culturing technique. Say for example, you want to use only cellulolytic organisms. So, you amend the culture media with the cellulose content. So, like that you have to also think about the primary screening strategies, learn about all primary screening strategies, also focus on secondary screening strategies, so that you know for which product what is the screening method you use, like enrichment culturing technique, etc. Not only that, even in strain development, understand the complete biochemistry of the organism that is screened and isolated, and the various auxotrophic mutants, how you use it for the strain development, what are the mutational studies that you use for development of a industrial organism. You have to also study strain improvement strategies, strain improvement methods, both at biochemical level and also at genetic level. Then in your syllabus, it is also there types of fermentation. Fermentation processes like surface fermentation, submerged and solid state fermentations. Of course, you will also talk about fed batch, etc. So, type of fermenters, types of fermentation. When you talk about fermenter design you learn, and all the accessories of a fermenter, you try to know the importance of baffle, spargers. So, all the accessories of the fermenter you learn it, and also types of fermenters for which product, what type of fermenter is better used. And also the type of fermentation processes, as I mentioned, for citric acid production, whether it is surface fermentation useful, solid state fermentations for which enzymes you use. So, identify the differences between surface, submerged, solid state, and you try to make a notes on this. It will be useful for your entrance exam preparation. And up and downstreaming processes, especially downstreaming processes and recovery processes are very important. For every product you use different techniques. So, list it out all of these downstreaming processes. And in industrial microbiology, once you understand what is the once you optimize the media conditions, again in optimization of media conditions, raw materials that are used in industry, list out all raw materials used in industry, because that plays a very important role. If you say it is alcohol production, you go for molasses, and you also go for alternatives like constip liquor and all. but you should know what is the primary raw material which is abundantly available you will be choosing. So, when you are focusing on this fermentation processes, selecting from microorganisms to use of raw materials and optimizing fermentative conditions and using a proper fermentation type and recovering the product is very important. Say for example, for alcohol, for distillation process, etc. So, for every product, downstreaming also important, and list it out. It is better you have a nice tabular form where you will write the name of the organism, what is the product it is producing, what is the fermentation process that is recommended, and is there any peculiar or specific or unique fermentative procedure is there, you highlight it, and also you write the applications of that product and also mention what is the downstreaming process that is being used for commercial production. Okay. So, these things you have to be well versed, that is completely about the type of product and the fermentative conditions and the process of fermentation. Once you understand these preliminary components, and try learning about different microbial products, and in the process of the metabolism, it produces these products, whether it is a primary product or a secondary product also you should know, because continuous culturing, fed batch culturing, all those to use, you should know which phase of its metabolism you have to concentrate, so that you can manipulate the strain and you can exactly make conditions suitable for the strain to produce the desired product. So, here you, in your syllabus it is given ethanol production, beer production, wine production. So, almost all are distilled and non-distilled liquors. So, in this area list out distilled liquors, non-distilled liquors, and ethanol producing organisms, not only yeast, even the alternatives like bacteria like 𝗬𝗏𝗔𝗧 mobilis for ethanol production, and for beer production Saccharomyces 𝗂𝗽𝗃𝗃. So, for wine production there is a specific name of yeast is being mentioned, even that you try to remember, and for all these ethanolic products you make a flow chart for all alcoholic products, and you have vitamins and amino acids and antibiotics also. So, for tetracyclines which strain is used, which strain of Streptomyces is used, and for penicillin you know that Penicillium notatum and all, Chrysogenum. So, you please list out all the products and the names of the organisms and understand the fermentative production, all the steps involved, especially from wine and beer you will get more number of possible questions, and the use of hops, all these things you have to mention and you have to make a note of it. Better is for every unit you write down the keywords, you make important terminology that is being used in that. The term pitching is also frequently used with reference to inoculum development. So, don't miss out any word which is unique to the process of fermentation. Okay, and you have even the modified fermentative products like you have microbial products, genetically modified organisms are also used for especially with reference to insulin production. You have this in your syllabus, that is microbial products from genetically modified organisms. So, in industrial microbiology, in industry what happens is not only that, you have this QA, QC and GLP and GMP, quality assurance, quality control, and good lab practices, good manufacturing practices. This is very important. No batch of product will come out of the industry without following these strategies, that is QA, QC and all. So, departments are mentioned there. So, please look into it, those terms, and patents and IP are also because this is industrial microbiology, any product that is used for large scale production, who patented it and all should be known. So, patent and IP are some questions related to that also, you please prepare. And I am here with some questions over here, like the first question is, the following statement is true statement with reference to alcohol production. So, molasses is the raw material used for alcohol production, and 𝗬𝗏𝗔𝗧 mobilis is used in alcohol production, distillation process is used in for alcohol recovery. So if you look into it, it's very clear that all the statements are true for alcohol production. With reference to industrial microbiology, you have fermented, previous question please. So molasses is the raw material used for production. Zymomonas mobilis is a bacterium which is used for alcohol production. Distillation is used for the process of recovery. So not only the regular organism, whatever the alternative producing organisms also you remember, because if they ask for true or false statements, you need to know alternatives also. Then the next question is, which of the following is not a common source of probiotics? So here all probiotic products are given, like sauerkraut, probiotic supplements, raw meat, kombucha and all. But it's very clear raw meat is not a probiotic product. Genetically modified organisms used for insulin productions are primarily derived from which species? So here given human, bacteria, yeast, animal. So with reference to this, yeast is been taken as the correct choice. So next topic is immunology and chemotherapy.
So coming to the immunology and chemotherapy. So there are two subjects that are clubbed here in this unit, and what I suggest you is, when you are studying immunology, it is all study about immune system. Don't think that it is just about antigens and antibodies. If you study, that is enough. So what I suggest you is, you take a big white sheet paper and then write from the entry of antigen inside the body, what happens inside the body. That will cover most of your topics that are there. That is what we call it as concept mapping. Okay. Now just in a nutshell to tell, suppose what happens when an antigen enters inside the body. So when an antigen enters inside the body defends against it. So it tries to protect the body from the antigen. So you have innate defense barriers that are there. Okay. So you will study about all the innate defense mechanisms that are there when you study the first part of it. So map it, antigen trying to enter into the body, all innate defense barriers, they try to stop the antigen from entering the body. Now the antigen has entered inside the body, it has crossed the innate defense barriers. So it is always a fight between the immune system and the antigen that has entered inside the body. If it has crossed the innate defense barriers and entered inside the body, it tries to colonize inside the body. But the immune system will not keep quiet at that time also. So the acquired immunity starts playing a role. So when the acquired immunity starts playing a role, then you will study about all the acquired immunity, like what is active immunity, how active immunity takes place inside the body, what is passive immunity. So you will cover all that aspects of types of immunity in that. Okay. Now, okay, it has entered inside the body, crossed innate defense barriers, then acquired immunity started coming into role. So while studying all these things, we will study about there are certain cells that are present inside our body which start attacking the antigen when it enters inside the body. We call those cells as phagocytic cells. Remember, this is very important. So the phagocyte, what are the different phagocytic cells that goes and eats the antigen or clears the antigen from the body? Remember, neutrophils, macrophages. So what is the phagocytic mechanism, how it engulfs the foreign antigen and all other things. Okay. Apart from that, remember one more very important terminology, antigen presenting cells. Okay. There are some specific types of antigen presenting cells inside the body which we call them as professional and non-professional. So when it comes to professional antigen presenting cells, remember the examples: B cells, macrophages, dendritic cells. Remember, don't forget these three names: B cells, macrophages, dendritic cells. They process the antigen, they engulf the antigen, they process the antigen into smaller peptides and display it with the help of MHC. Again, one more topic in that is covered when you are doing this concept mapping. What is that concept? Here you are studying about MHC. What does MHC stands for? Major histocompatibility complex. Okay. So I always tell the students to understand the name of the word major, histo, histocompatibility complex, learn it like that, which plays a major role in matching the tissues. Generally a graft will not be accepted, right? So like that, when there is a kidney transplant or when there is a liver transplant, there will be acute rejection. Not only that, it's a simple blood grouping also, transfusion also, there will be an acute and chronic rejections that takes place. So map it like that. So now the antigen presenting cell has presented the antigen with the help of MHC. There you will study about major histocompatibility complex, what it is, MHC class 1, MHC class 2, and when the antigen is presented by MHC class 1, then to what type of immune response takes place. It is cell mediated immune response. You will study that again, cell mediated immune responses. If the antigen is presented by MHC class 2, what type of immune responses takes place? Humoral immune responses. Sometimes if it is mediated by T helper cells and cytokines, then it is another thing. So like that, you may make a concept mapping and try to interconnect, because immunology is not that subject where you study it in bits and pieces. It is a subject that has to studied in continuation, then only you will understand the proper mechanism of immunology, what actually the mechanism is, and any type of question that can be asked in immunology, you will be able to answer it, whether it is a logical question or an analytical question or a higher order thinking skill, HOTS question also, you will be able to answer it when you do the concept mapping in immunology. Okay. So the first basic part of immune system, types of immunity, and how the phagocytic cells plays an important role, and what are antigen presenting cells, MHC, everything is done here. Now once it is done, from B cells antibodies will be produced, from T cells cytotoxic T lymphocytes or cell mediated immune responses will take place. Now the antibodies are produced and they are released into the circulation. See it like a movie, then you will understand, appreciate the immunology more. Okay. So when antibodies are produced, what happens is antibodies will be released into the circulation, and wherever they come in contact with an antigen, they come and bind with the antigen, okay, forming an antigen antibody complex. So with that, the part 1 of story of immunology will be done. Now what is this antigen antibody reaction? There you study here the different types of antigen antibody reactions also. Make a map again, a mind map of antigen antibody, different types of antigen antibody reactions. Categorize them. The antigen antibody reactions in vivo and in vitro, or label them as secondary labeled antigen antibody reactions. These labeled antigen antibody reactions is very, very important in examination point of view. I don't say that the primary antigen antibody reactions are not important, but these play a very, 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 very important role in diagnostic part of it. Okay. So in primary, agglutination, precipitation, neutralization and complement fixation. In the second part of it, the labeled ELISA, radioimmunoassay, immunofluorescence, flow cytometry, Western blotting. So remember the terms and for what they are used for. Okay. So that antigen antibody complex is formed inside the body. So there is what happens when an antigen antibody complex is formed inside the body. It has to be removed from the body. If it is not removed from the body, what happens? If it is not removed from the body, it results in autoimmune disorders, trapping of antigen antibody complexes. There you will study the autoimmunity. Okay. So different types of autoimmune disorders like organ specific autoimmune disorders, systemic autoimmune disorders. So list out what are the autoimmune disorders that are there. Organ specific, thyroiditis is an autoimmune, organ specific autoimmune disorder. See the name, organ specific, systemic, it is throughout the body. Rheumatoid arthritis is a systemic autoimmune disorder. Systemic lupus erythematosus is a systemic autoimmune disorder. Okay. And glomerulonephritis, it is an organ specific autoimmune disorder. It is easy to categorize when you understand the mechanism of it. Okay. Now, how these antigen antibody complexes are cleared from the body? They are cleared from the body by an effector mechanism inside the body, which is called as complement system. There you will study about the complement system, classical pathway, alternate pathway, lectin pathway and other things. Okay. We understood what happens if antigen antibody complexes are not removed. You studied about autoimmune disorders. Now, what is the other phase of immune system? What happens if immune system is hyperactive? If it is aggravated, it results in hypersensitive reactions. that is also dangerous to the body. Okay. So, 4 types of hypersensitive reactions, list them 1, 2, 3 and 4 types of hypersensitive reactions, and remember them as what is the mechanism of hypersensitive type 1 anaphylactic reaction, what is type 2, what is type 3 immune complex mediated, so complement mediated, like the delayed type. So, remember it with respect to the name of the mechanism that it takes place. Okay. With that, the basic part of immunology you have covered just by mapping it in one A4 size paper, writing about everything and tagging it with the side reactions, whatever side lines that are there which are specific to that. Okay. Then there are some more specific aspects that are there in immunology, like cancer biology, tumor immunology. Okay. What is this tumor immunology? What happens when there is a tumor inside the body? Will it act like the same the way the antigen is acting against antigen, or will it act in a different way? It will act in the same way, but there is a slight deviations that are there. That mechanism part of it you study it in tumor immunology. Okay. And how vaccines are prepared and all other things you will be studying here in the immunology part of it. So, there is one more concept called as tolerance. What do you mean by a tolerance? What is a central tolerance? What is a peripheral tolerance? When the immune system stop reacting, sometimes the immune system stops reacting to the antigen. So, what is that condition called as? What is central? What is peripheral? Everything you study it in detail here. Now, this is part 1, chemotherapy. The second part is chemotherapy. In chemotherapy, you will study about different types of antibiotics, different types of antiviral agents. Just now we have seen in one previous bit why antibiotics act on bacteria, antivirals act on viruses, antifungals. So, like that different terminologies are given. Don't get messed up with every terminology and combine everything. Okay. And we will discuss about what are different types of antibiotics, what is the mode of action of different types of antibiotics, cell wall inhibitors, penicillin, cell membrane inhibitors, polymyxin, then protein synthesis inhibitors, streptomycin, tetracycline, antimetabolites, sulfa drugs. So, like that you make a categorization and you study. These questions will be coming in your examinations regarding the antibiotics. Okay. And different types of microbial assays will be there. So, you study about different types of microbiological assays also, like how to do Kirby-Bar method, then giant plate method. So, all those methods, Stokes method. So, all these methods you make a list of it, and tube dilution assays, how minimum inhibitory concentration is measured, all those things you please make a note of it. Okay. Next, coming to some of the model questions that are there in immunology, examples of professional antigen presenting cells. Just now I told you it is very, very important, remember that they express class 2 MHC. So, the options given are dendritic cells, macrophages and other things. So, all the above is the answer. The only immunoglobulin that crosses the placenta. When you are studying antigen and antibody, you will study about the details of antibody structure and the functions of antibody. So, it is IgG antibody is the only antibody that crosses the placenta. And what is the mode of action of penicillin? It is a peptidoglycan inhibitor. So, like that you make the model questions for each and every aspect. The next is food microbiology.
So, the next component of our microbiology is food. This is very, very important because when you talk about food products, in food microbiology generally we discuss about food spoilage, what are the organisms which are responsible for food spoilage, and what is the food substrate. Say for example, it can be carbohydrate, it can be a lipid, or the biochemistry of the food also you should know, because carbohydrates, they are getting fermented and they are getting spoiled. So, if it is lipid, lipolysis, protein, proteolysis. So, the content of food, what makes it to spoil and what is the role of microbe for such spoilage is important, because these microbes, they are capable of producing those enzymes which are actually meant for such biochemical change. So, understand the principle of food spoilage and also know about what are the microorganisms which do cause this kind of spoilage. With reference to milk, fresh foods and canned foods, these are the examples that are given in your syllabus. So, bacteriological examinations of these foods, like SCP, viable count, all these things you have to look into it. And with reference to dairy microbiology, what are the different dairy products and what are the microbial products of milk, what are the fermented products you should know. And coming to the preservation methods both, even though chemical methods are popular, you also should know what are the chemical preservatives like benzoic acid, sulfur dioxide, propionic acid, etc. And also know about their for which type of spoilage, what type of preservative you use. So make a list again for the type of food, what is the food preservator you are using, preservative method you are using, and what are all the principles of spoilage. And here you have a very important aspect, that is food safety, hazards and risks, current and future implications concerning food safety. Please look into the newspapers and find out what are the latest advancements that are seen in food safety protocols. Then probiotics is one very important area, because everybody is talking about probiotics, prebiotics, postbiotics. Clearly know about these three terms, probiotics, prebiotics, postbiotics, because their significance on human beings and animals. You need to understand what are the probiotic foods and in what way they are nutritive and all. So with reference to food microbiology, emphasize on spoilage, preservation, food poisonings, and what are the diseases that are associated with foodborne illness, all that you need to know it. So here are some questions, model questions. Scanning is also called. So this is a direct question. You can you know what is blanching, what is pasteurization, what is sterilization. So the correct answer is apparatization. So this is the answer. Next question is, prebiotic foods are primarily a source of. It is fiber. The next question please. So which of the following statements refers to fermented foods? Again, I tell you, whenever such statements are given, read the sentence completely and give the correct answer. In this case, it is all about fermented foods. Fermented food is made from cabbage, is referred to be kimchi. Popular fermented soybean is natto from Japan. Traditional fermented dairy product is yogurt. So all the statements are correct. Here also I again mention with reference to fermented foods. What is the source of fermented food? From which country it is so popular? All that you have to keep in mind, and the name of that fermented food also.
So we'll just quickly go through the other things also that are mentioned in the syllabus, that is molecular biology and microbial genetics. In this molecular biology and microbial genetics, as you are very familiar with it, so here you will be studying about the structure of DNA, the transcription and translation processes that takes place, and what is genetic code, and what are the different cloning and expression vectors that are there, what are the different molecular methods that are mentioned like PCR, gene chips, microarrays, and what is a DNA sequencing, so what is gene therapy. So all the aspects, please try to cover every part of it in molecular biology and microbial genetics. In the last part of it, so some aspects of bioinformatics is also added, which includes databases, then sequence and structure analysis of DNA and proteins, primary designing, protein engineering, and drug designing, and all other things. Okay, let us quickly go through the questions, model questions. So what is nested PCR involves? Nested PCR always involves 2 rounds of amplification using 2 different primer sets. So go through what is the meaning of a nested PCR. Then the next is the key feature of conjugative plasmid in bacterial conjugation is their ability to. What is its ability? It transfers the genes between the different species. And what are transposable elements? Please have a thorough knowledge of what the transposable elements are. So transposable elements are here among the options that are given, it includes all the above. Then let's quickly go through about the environmental and agricultural microbiology and wrap up the sessions.
So this agricultural microbiology, we have the microbiology of water, and sewage as the first topic. Of course, the ecological significance is all about referring to environmental and agricultural microbiology. The role of microorganisms in nutrient cyclings, because these cycles like phosphate solubilization, nitrogen metabolism, all these are very important. Focus some light on this nitrogen cycle and other biogeochemical cycles. And biofertilizers, biopesticides, please list it out all the examples. And biodegradation of pollutants, plant microbe interactions, PGPRs are very important. Learn characteristics of it and also make a list of examples of it. And animal microbe interactions, rumen microbiology, and the production of energy from agricultural and domestic waste. So this is the syllabus given to you. So read a proper textbook for this. We have so many textbooks available, even the general microbiology books like 𝗪𝗬 or even the other textbooks, you can refer it for these fundamental topics. Some questions are given over here, that is the biodegradation of pollutants, which factors influence. It is both temperature, humidity, soil texture. Then the next model question given over here is with reference to sewage treatment, that is in the activated sludge process, the term activate sludge refers to. Again, when all these statements are there, you have to be carefully reading and picking out the correct answer. The microbial biomass that is suspended in the treatment tank is actually the term which refers to activated sludge. Then the next example question is the primary function of rumen microbiota. This is from rumen microbiology. So, the digestion of carbohydrates is the right answer. There is a last 2 units left out. We will quickly focus on it.
So, when coming to the biochemistry and techniques, here we need to study about the structure, classifications of different types of carbohydrates, proteins, amino acids, and study about the enzyme nomenclature, study the different types of colorimetry, study the different types of spectroscopy, and also study about the different types of chromatographic techniques that are there in the syllabus. So, while quickly going through the syllabus of biochemistry, we have also 2 more topics that are there, that is medical microbiology.
So and in medical microbiology, the clinical significance is very important. Both bacteriology as well as virology you emphasize, because you have got different sources for infection like airborne, waterborne, foodborne, insectborne, zoonotic, all examples you look into it, because epidemiology, clinical significance, pathogenicity, how it is treated, all that is very important in medical microbiology. And even the pathogenicity factors, virulence factors, you focus on it, because this topic is very important. We have seen COVID pandemic recently. So, even from COVID, coronavirus also, what are the vaccines, where are they produced, what is the principle involved in production, all that you make a note of it. So, quickly we will see 1 or 2 questions from medical microbiology. So, here is the question, choose the incorrect pair. So, again incorrect pair means you have to see both left hand side, right hand side, whether it is given proper combination or not. Filariasis, Wuchereria bancrofti, malaria, Plasmodium toxoplasmosis by Gonidae, Giardiasis by Entamoeba. Amoebiasis is caused by Entamoeba histolytica. So, the wrong answer, the right answer goes to 4. Example of opportunistic mycosis, here is Candida is the typical example. All other 3 are dermatomycosis examples. Then the correct combination, VDRL test, RT-PCR, Widal, Mantox. This is diagnosis, because this is very important component of your syllabus. Here RT-PCR, everybody knows that we have used recently for coronavirus diagnosis. So from this paper, microbiology, for this degree college lecturers preparation, whatever for the posts or in residential institutions you are preparing. So, wish you all the best. We tried our level best to cover almost all the syllabus, but it is very important to have time management. You have a very, I think you have time for preparation, a lot, enough time for each of these units, and prepare well. I wish you all very best for your preparation, and also I am sure you will come out with flying colors.
All the best to all of you, and what I suggest you is you give equal importance to all the subjects or all the topics that are mentioned in the syllabus. Don't leave any aspect of it. Read environmental, food, industrial, medical microbiology, biochemistry, molecular biology, immunology, chemotherapy, general microbiology and virology. So, all the topics that are mentioned you cover equally for all the subjects. Take time, make specific timetable, allot specific time for each topic and prepare. I am sure when you, as madam told, I am sure when you do with the proper time management, you will definitely clear any entrance examinations, and I wish you all the best and good luck for your entrance examination. Good luck to all of you.
Thank you.
Thank you.
Thank you. Good luck.